Despite advances in medicine, cancer remains one of the most frightening diagnoses a patient can receive. What makes it so difficult to cure is that cancer characteristics vary from patient to patient, so there is no single treatment to cure all types of cancer. Leukemia is a cancer that originates in the bone marrow and it is caused by rapid production of abnormal white blood cells. Bone marrow is also home of blood-producing hematopoietic stem cells that can differentiate to all types of blood cells. The process of proliferation and differentiation is continually happening, but when mutations occur in genes that are involved in controlling this process, we have uncontrolled division and block differentiation, rising to leukemia. Once leukemia strikes, leukemic blast cells accumulate in the bone marrow, taking up all the available space and resources, and outcompete the normal hematopoietic stem cells that are vital for healthy blood production. So these patients suffer from blood failure. They can also enter into the blood circulation, creating more serious complications related to abnormal white blood cells. Treatment that is commonly used is chemotherapy, that is a combination of drugs that destroys rapidly dividing cells, but as a side effect, these drugs also kill some healthy cells, including hematopoietic stem cells. Therefore, we need more innovative approaches and individualized treatments to treat patients and minimize any side effects. New treatments should be able to only target the cancer cells, leaving the healthy blood-forming cells intact. A limitation that exists in studying these leukemic cells is that they don't grow and retain their cellular functionalities when grown in a regular lab dish, so it's really hard for scientists to test novel treatments on them. To overcome this problem, we are investigating whether we can grow these cells in a condition that mimics the bone marrow environment of cells rather than regular petri dishes. Stem cells live in a dynamic and specialized microenvironment called the stem cell niches. Extracellular matrix is an integral part of it as it can directly bind to different cells using surface receptors. Both the structural supports of ECM and signals that cells receive from them are essential for the proper function of cells. To mimic the natural tumor microenvironment, our group employed a system that involves seeding AML cells on engineered matrix constructs that are incorporated with other ECM components to define a favorable environment for these cells to live in. This approach allows us to study the way cells interact with their ECM and gain a broader understanding of how each of these components contribute to cell survival, proliferation, differentiation. Working in 96-well microtiter plates that permit experiment miniaturization and increased throughput, cells are seeded in the matrices and then recovered afterwards to examine how they responded to the matrix. Currently, we employ a destructive approach to remove the cells from these matrices. This method is laborious, expensive, and operator-dependent. My LURA summer project was to optimize this step to find a more efficient and non-destructive way to assess the cell quantity and cell behavior. In the proposed method, after seeding the cells on the scaffolds, we add a dye called Alamar blue that stains the cells that are alive and metabolically active. We then used a new screening technique with a microplate reader machine and tested whether addition of collagen to regular 96-well plates interferes with the results obtained by the machine, and if so, whether the reading can be calibrated accordingly. This machine excites the sample with a particular wavelength and can detect fluorescent light that was emitted by the sample. This gives us valuable information regarding the quantity of live cells that are supported by the scaffold. This is an ultra-fast, non-destructive screening technique that allows high-throughput screening of the scaffolds when they're tested with different conditions. We have found that interference occurs in film as we increase the thickness of matrices, so to optimize reading by microplate reader and minimize any interference, it is suggested to seed the cells on the range of 25 to 100 microliters of collagen. Additionally, to read the number of cells, we can add a control with collagen film to create a baseline to then calculate the true cell number. Also, it was found that ideal seeding density is 25,000 cells per well, as we see most other significance when compared to our control. Upon successful establishment of a culture system, we can employ it in the development of a high-throughput drug screening technique in which annotated drug libraries will be screened for potential new sources of treatment. Moreover, it can serve to use more personalized screening of patient samples to tailor a specific treatment for them to minimize their potential side effects. Thank you.